Okay, so actually if I go back one slide, this is moving into the laws of inheritance, section 10.3. We did all the review stuff in lab last week. And we said that the first law of inheritance is Mendel's law of dominance. We said this in lab last week. Basically, this says that in a heterozygote, one trait will conceal the presence of the other trait of the same character. So we looked at color and shape of corn kernels in lab last week. We've been perpetually looking at flower color in pea plants, right? So if we have purple flowers on a pea plant, purple is dominant to white. And so as long as that plant has at least one dominant allele, it's going to show purple color and the white's going to be masked. That's the law of dominance. And this is what you were testing with your corn kernels in lab. You were testing to see, is your corn ear following Mendel's law of dominance, or is something else going on? Today we're going to get to that something else. Okay. We're also going to, this is sort of awkward because I didn't do that chapter 9 yet. We're also going to look at Mendel's law of segregation, and we're going to do this kind of how Mendel did it. Remember I said that Mendel was shooting in the dark, and we hadn't discovered, genes or chromosomes or mitosis or meiosis yet. And so he came to his law of segregation without knowing anything about mitosis and meiosis, how gametes are formed. He just said that there are these heritable factors, and they get passed on so that paired genes segregate equally into gametes, and an offspring has an equal likelihood of inheriting either heritable factor. We now know that each individual in any population of any organism has 2 alleles for every given gene, right? And those alleles might be the same. You could be homozygous, right? Or they might be different. You could be heterozygous. And it doesn't really matter, because what the law of segregation says is that both of those alleles, regardless of what they are, they have an equal chance of going into any gamete that individual produces. So this diagram here is showing you meiosis, which we haven't talked about yet, in which a diploid parent has 2 alleles carrying some gene S for seed shape, maybe, and then those 2 are going to duplicate into a pair of sister chromatids during the S phase of the cell cycle, right? So here we have duplicated sister chromatids. And in meiosis, there's a series of 2 divisions. In the first division, the homologous pair of chromosomes separates so that the genetic information that came from one parent goes to one side, and the information from the other parent goes to the other side. So here, the 2 alleles are going into opposite cells. And in meiosis, we have a second cell division, and that's where the sister chromatids come apart. So we make 4 gametes, and each one of those gametes has an equally likely chance of receiving any of the genetic information in the parent. All this is really saying is that how these chromosomes divide when gametes are being formed is chance. It's totally random. There's not something specific that always moves one allele to one side or one allele to the other side during cell division. It's just chance, okay? And so any one of these gametes could get the dominant allele or the recessive allele, and it's just luck that they get what they get. Okay? And that they must separate. The homologous chromosomes, the 2 alleles for any given gene, do not stay together. They separate during meiosis. Okay. Is that too confusing? You guys are looking at me like, it'll make more sense. We'll come back to it. It does all seem kind of similar. It's because it's all related. It's all really similar because it's all really related. These similar processes are happening in the same organisms. All right. I have faith in you. Mendel's, I think this is the last law, right? Yeah. Mendel's law of independent assortment says that the genes don't influence each other. So when we looked at our seed color and seed shape in our corn ears last week, right? The color is controlled by one gene and the shape is controlled by a different gene. Those genes are likely on different chromosomes, right? And basically what the law of independent assortment says is that the genes don't influence each other. So when the chromosomes are dividing and genetic information is getting allocated into gametes to make egg and sperm, or pollen and ova, there is no force or interaction between those genes that cause them to be inherited as a set. It's just random chance, again, okay? So if we have two pairs of chromosomes here, maybe one of these carries seed color and one of them carries seed shape, how the seed color chromosomes line up has no impact on how the seed shape chromosomes line up. So here in this example, the two blue chromosomes are on the left-hand side, the two red ones are on the right. It could easily be that they're opposite, okay? And by the time you get up to humans with 46 chromosomes, right, all the possible ways that those chromosomes could line up and separate, it's almost infinite, right? It's very, very, very high. And so because of that, our gametes are unique, and they're unique because of random chance. There's some caveats to the law of independent assortment, which is that this only occurs if genes are on different chromosomes, because many genes are on the same chromosome. And if they're all on the same chromosome, then they're going to go together as a set, right? And the way that this happens is due to the random alignment of chromosomes during metaphase 1 of meiosis. And again, we'll get meiosis on the flip side. Okay. And I'm going to just leave these here. For you can study this diagram if you want. This is just showing you two potential pathways this same parent cell could take to get to these 4 gametes, which are different than those 4 gametes. And just have that there for you guys to look at if you want to try to sort this out. I also have pipe cleaner chromosomes. And I know that sounds really ridiculous, but some people really love the pipe cleaner chromosomes. And if you want to check out one of my chromosome sets and practice mitosis and meiosis with a chromosome set, I'm happy to sort of sit down and walk you through that. It can be really helpful for some of you who are very visual learners. Okay. We also looked at dihybrid crosses, right? So I want to just really formally and quickly go through dihybrid crosses. A dihybrid is any plant differing in 2 characters in the F1 generation. So in this P example, we have a difference between seed shape and seed color. In our corn example last week in lab, we had seed shape and seed color, right? In pea plants, spherical yellow seeds is the most dominant condition. So spherical shape or round shape is dominant to wrinkly shape, and yellow is dominant to green. Okay, so if we have a true breeding parent plant that is homozygous dominant, it's going to have spherical yellow seeds, and it's going to be big S for seed shape, and big Y for yellow color. Okay, if we breed that with a true breeding homozygous recessive individual, the recessive characteristics are to be wrinkled and green seeded, right? So the phenotype would be to have wrinkly green seeds. That means that they're going to be little s for seed shape, wrinkly, and little y for seed color, green. Okay. So I think I went around and did this for most of you. Tommy actually, where are you at? Tommy has a great little square that he did instead. But one of the things that we need to do when we're going to perform this cross is, and to move to the next generation, is to be able to predict what's going to go into the gametes. So just like in our monohybrid cross, when we cross our dihybrid individuals, our F1 generation is all going to be heterozygous, right? So all of the individuals in that F1 generation are going to be heterozygous for both shape and color. Big S, little s, big Y, little y. They're all going to be yellow and round, 100% yellow and round, right? If we want to make them the F2 generation, we're going to allow the seed to produce a plant that self-fertilizes, we have to figure out what are all of the possible combinations of those 4 letters. And every time that parent plant makes a gamete, it is going to give its offspring both a shape gene and a color gene, right? So if we're going to make 4 separate gametes, each gamete is going to have both an S and a Y in it, because it's going to give all of its offspring an allele for color and an allele for shape. Does that make sense? So you can do a quick little sorting and just say, okay, this S could go with this Y, and we get big S, big Y. That same S could go with this little y over here, we get big S, little y. The little s could go with big Y, or the little s could go with little y. So we have 4 possible gametes for our F2 generation. Because it's going to self-fertilize, we would just set up our Punnett square with those gametes on both the top axis and the side axis of our Punnett square. So this is what our F2 Punnett square looks like. It's very analogous to what you did for corn. It's also analogous to what you did in the second part of the corn lab for the epigenetic with the expression gene. It's always pretty much the same because we have heterozygous individual self-pollinating with a heterozygous individual, and then we look at our results. This is nice because it's color and shape coded, and we can see all of our outcomes. We can also see that we get some recombinant phenotypes. And what that means is we get some new phenotypes that didn't exist in our parent or F1 generation, right? In our parent generation, we had this and this, right? Now, all of a sudden, we've added in green round seeds and yellow wrinkled seeds. We have these new outcomes because of how we've reshuffled the genetic information. Okay. When you go through and count up this Punnett square, you would determine that there are 9 separate genotypes and that there are 4 phenotypes. And in our complete dominance cross for an F2, we're always going to have a 9:3 to 3:1 ratio in our F2 generation. The most dominant trait will have 9 in the phenotype. That's our yellow and round. The 2 intermediates, the green wrinkled and the green round and the yellow wrinkled, will each have 3 out of 16, and then the rarest here will be 1 out of 16. Okay, and you guys went through and counted that up in lab, so hopefully that feels familiar. Does anybody have any questions on that after doing it? Okay. So now I want to move on and look at some extensions and some other pieces that go along with Mendel's laws of inheritance. And the first is the notion of test crosses. And test crosses are becoming increasingly rare as we have more and more powerful genetic tools available at cheaper and cheaper costs. But a test cross is basically a way to determine whether an individual is either homozygous or heterozygous. So if you have an individual that shows a dominant trait, you don't know if it's homozygous or heterozygous. You just know that it has the dominant phenotype, right? One way to determine whether it's heterozygous or homozygous is to cross it with a homozygous recessive individual. So obviously we're not going to run around and like forcibly breed humans together. But this has been used a lot in, like, say, dog breeding or horse breeding. They want to determine what they think that one particular parent has for genetics and who they might want to match that parent with in order to get a particular outcome. Okay. So we could say that we've got some round green seeds, and in this case, shape round is dominant. And we want to know if this individual is big S, homozygous dominant for the shape allele, or little s, homozygous recessive. So we can cross this individual with a homozygous recessive individual, right? And if we look, our 2 crosses would be our dominant allele next to our homozygous dominant versus our homozygous recessive, or our heterozygote next to the homozygous recessive. The outcomes are different, right? If 100% of the offspring have the dominant trait, then the parent must have been homozygous dominant. If we get a 50-50 split, half of the offspring have the dominant trait and half have the recessive trait, then the parent had to have been a heterozygote, right? The fewer number of potential combinations there are, the easier this is to solve, right? But you need sample size. If you're looking at something like a litter of puppies, so maybe we want to look at black labs versus chocolate labs, right? If you only have 2 or 3 puppies, you may not have actually had enough puppies to know, right? Just because they're all black doesn't mean, just means we didn't get a brown combination. So you might have to have a couple of litters before you could try to figure this out. So if you want to look at if you want to do a practice test cross, you could answer this question. The answer is there. I will flip through it so you can figure it out on your own. But know that we don't do this as often now, that we often just use genetic analysis.
No, I know it says like 1 out of 4 should be like whatever trait, but like no one offspring change the other. They're independent, right?
Yeah, the offspring are independent.
Okay, so
right.
Yeah.
Right. It's just that you have to have enough of a sample size. Because if you only have 4 puppies, that doesn't mean that you've met all possible combinations of genetic material in producing those 4 puppies, right? It's a lot easier with plants. We have 1,000 seeds, and we can like have some sample size to figure that out. Okay. The next thing I want to talk about just briefly, hold on. I feel like, oh yeah, there's just one slide. We're going to come back to most of this conversation. I want to give you a very broad and general notion of sex determination in humans, so we can talk about those particular genes that are found on the sex chromosomes. Okay, and so in humans, sex determination initiates in the chromosomes, and so it's in the chromosomes that we determine ultimately what internal physiology is going to form, and that internal physiology dictates hormones and ultimately external physiology or external anatomy. Okay, so in humans we have a total of 46 chromosomes. 44 of those chromosomes are called autosomes, and those 44 autosomes code for most of the things about the body. Okay? The last pair of chromosomes, either XY or XX, are the sex chromosomes. And it is the presence or absence of that Y that controls maleness. So individuals who are XY, if they follow the most typical common pathway, will develop into individuals who are male. And individuals who are XX will typically follow a pathway to develop into a female. So when we cross 2 individuals, one of those individuals has 22 chromosomes plus an XX, and the other individual is 22 plus XY. And how the Y and X come together will determine the sex chromosomes for the next generation. Right? So here we have this XY individual, and that XY individual can make offspring, make gametes that are either 22 autosomes plus an X could go into the sperm, or 22 autosomes plus a Y can go into the sperm. This XX individual over here is always going to put an X in the egg. It could be one X or the other X, but every egg gets an X, right? So ultimately, the offspring will have 1 or 2 X's and 1 or 0 Y's. Does that make sense? Okay. This is important to point out as something separate, because these are the X and Y chromosomes. This is X, this is Y. Y carries a few genes, not very many. Okay? The X chromosome is actually quite large. and carries many genes. Not all of those genes are at all related to sexual development of the individual. Okay, the Y chromosome carries a really important gene known as SRY that regulates testicle development. The X chromosome carries all sorts of genes that do other functions, have nothing to do with sexual development. Okay, but because XX individuals have 2 X's and XY individuals have 1, if there is a disease or something that is in the X chromosome, then XY individuals are more likely to get it, right? Or something that is a recessive trait. XY individuals are more likely to express it. So X has many genes, Y has few genes, and anytime we're talking about XY chromosomes, we refer to the XY individuals as being hemizygous. So we had homozygous, we had heterozygous. Homozygous means you have 2 copies of the same allele. Heterozygous means you have 2 different alleles. Hemizygous means you only have 1 allele, and you can only ever have 1, right? So for an XY individual, will only ever have 1 copy of the X gene. So whatever it gets on the X chromosome, whatever variant of the gene it picks up, that's what's going to be expressed. There is no dominance or recessiveness because there's nothing to mask a trait, right? The X is just expressed as it is. Okay? So XY individuals are hemizygous for anything that's found on the X chromosome. An organism that has a similar sex determination system to humans is the fruit fly. Fruit flies are the workhorse of genetic studies because they have some analogies to humans that allow us to ask questions with their genetic manipulations. And one of the chromosomes that is found on the sex chromosome, or one of the genes that is found on the sex chromosome in fruit flies is for eye color. And so the wild type, most common sort of native eye color is red, and red is dominant. But you can also have individuals that have white eyes. Okay? And whether you have red eyes or white eyes comes on the X chromosome. So we'll use fruit flies as our example here. So here we have a female fruit fly, an XX fruit fly. And this XX fruit fly is homozygous for the dominant trait. She carries 2 alleles for red eyes, right? If we mate this individual with this XY individual over here, this hemizygous individual is white-eyed. It has 1 copy of the white allele, and then it has the Y chromosome, right? So it's hemizygous. It doesn't have 2 alleles. It can't be homozygous or heterozygous, but it is white-eyed. It carries the recessive trait. Does that all make sense? Okay. So if we look at a cross for this individual, if we cross the recessive white-eyed allele and the Y chromosome with our 2 dominant X chromosomes, we find that all of the XX offspring here will be heterozygous, and all of the XY offspring will have red eyes, dominant trait. And that's because all of those XY offspring got Y from their XY parent, and they got this dominant X from their XX parent. Does that make sense? Okay. If you look at a different cross, here we have a homozygous white-eyed XX individual, 2 copies of the recessive allele, and we mate that with a hemizygous red-eyed XY individual. Then we end up with another strange pattern in which all of the XX individuals are heterozygous, and all of the XY individuals are hemizygous white-eyed, right? So when you do these crosses, or when you're given questions that ask you about X-linked traits, you have to remember that there's only one copy in the XY individual. And you have to make sure that the other chromosome is Y. Y doesn't carry any eye color, right? This chromosome is neither red-eyed nor white-eyed. It is male. It is Y, right? It's not carrying an allele to match up with what's on the X chromosome. Does that make sense? It is. There we go. So this leads to a lot of recessive X-linked phenotypes. And so in the human population, there are several genes that are known to occur on the X chromosome, and most XX individuals have the dominant phenotype because if they carry the recessive phenotype, they're likely to also have a dominant phenotype and just to be heterozygous, right? So most of the individuals who are affected by the recessive condition are XY individuals. And so one example is red-green color blindness. So this is just a colorblind test. You can tell what numbers are here. What's this one? It's hard, right? I could turn the lights off and it makes it a little bit easier. Is anybody in here colorblind that you know of? I keep waiting to meet an XX colorblind person, because I haven't yet. But it can run very tightly in families through the passed down through that X chromosome. Another example is hemophilia, male pattern baldness. There are a handful of traits that are known to be found, or characters known to be found on the X chromosome. Okay, so now this is still section 10.3. I thought other section might have been mislabeled then. Anyway, now what we're going to do is what I said we were going to do at the beginning, is actually start to look at some other ways we can apply the laws of inheritance. And so far we've been keeping it really simple. We've been looking at a very straightforward dominance recessiveness pattern, and most traits are not as simple as the examples we've gone through so far. So we're going to look at some more complicated examples. And before we do that, we're going to mention the chromosome theory of inheritance. And so once Mendel's work was rediscovered and we understood how mitosis and meiosis work, we had a better understanding of cellular mechanics, starting in the early 1900s. There's this modern synthesis of putting information from disparate locations together, and ultimately that led to the chromosome theory of inheritance. And so this biological theory basically just says that genes occupy specific locations, plural is loci, or single loci on chromosomes. And those chromosomes undergo segregation during meiosis, so they separate, right? And that happens independently. We mentioned the law of independent assortment, right? So the chromosome 1 is not dependent or impacting chromosome 2 in any way when gametes are made. And it is the chromosomes that account for observable inheritance patterns. Right? In all of Mendel's work, he was just talking about heritable factors. He didn't know what they were called. So this is just using our modern language to describe what we've already talked about. Okay. You guys are still writing that down? Good. I'm going to actually skip that review. So we've talked so far mostly about complete dominance, the case in which the presence of one allele will completely mask the other allele, like in our purple pea flowers. The dominant trait is going to show whether we have a homozygous dominant individual or a heterozygous individual. We only see the recessive when we have a homozygous recessive individual, right? That's been our underlying assumption, right? I always like to think that if Mendel had studied something else, like say snapdragons instead of peas, he would have never figured out any of the things that he figured out. And that's because he just had a really lucky pick of plants. He picked a plant that has a really discrete set of characters that are very clearly heritable by complete dominance. And so he wouldn't have gotten to his laws of inheritance if he hadn't studied the pea plant, because we have other ways of inheritance One of them is known as incomplete dominance. And incomplete dominance is what we find in snapdragons. And somebody asked me before about mixing, I think, and I said we'll get back to that. So incomplete dominance is where some alleles are neither dominant nor recessive. Okay. So if we start with a red snapdragon, red is technically our dominant, big R, and white is our recessive, little r, right? That red snapdragon is going to give all of its gametes a big R, and that white snapdragon will give all of its gametes a little r, right? If we perform the F1 cross of these two parents, our F1 generation is 100% pink. Not a red or white individual to be seen anywhere in that generation, okay? So all of the heterozygotes are an intermediate phenotype between the two parents. It's like they're blended, right? And that's because neither the red nor the white is completely dominant or completely recessive. Since it's incomplete dominance, they're both partially expressed. So you end up with this blended appearance. But if you take our F1 generation heterozygotes and you allow them to self-fertilize, just like we did in our complete dominance studies, so we have them making gametes, half of their gametes get a dominant allele and half of the gametes get a recessive allele, then you end up with an F2 generation where 1/4 of your individuals are red, 1/4 are white, and half are pink. Okay? Your genotype ratios are the same as in complete dominance, right? We have a 1:2:1 genotype ratio. Does everybody see that? I was supposed to ask you. This is my note down here. 1:2:1. So 1 big R, 2 big R little r's, and 1 little r. What is, go ahead.
How are the sample years of corn you keep getting wrong?
I don't know where the splotching is. I don't know where the splotching is coming from in those ears of corn. I need to have a conversation with who, like, I'm sure that all these different biological supply houses are getting their corn from the same person. Like, there's probably not that many labs breeding genetic corn. I need to, like, just track down where they're coming from and ask these questions. There's also transposons, jumping genes, and weird stuff that happens in corn that, yeah, I think they don't have things under full control. What is the phenotype ratio here? It's the same as the genotype, right? 1 red individual to 2 pink individuals to 1 white individual. Okay. So here our genotype and our phenotype ratios are the same because neither allele is completely dominant. Okay. Another case that is super common is that most genes don't just have 2 alleles. It's not like there's just a pink allele or a white allele, just a purple allele or a white allele. There's all sorts of different alleles, right? And because there's all sorts of different alleles out in the world's population, that means there's all sorts of different possible combinations, depending on which 2 each of your 2 parents had, right? So this increases the total number of possible phenotypes. And typically what we find is that when we have those multiple alleles, there's a hierarchy of dominance. So one allele is like the most dominant, and one allele is the most recessive. And then there's a continuum between where there's a hierarchy of how those alleles combine with each other. And so in this case, we have coat color and rabbit. And this is another one of those places where I feel like I need to tell you, I'm never going to ask you what allele combinations give you a dark gray rabbit, right? This is just an example to try to make this point. But the most dominant phenotype is dark gray, like this guy down here that's almost black, right? And you can get this almost black rabbit by having any allele set which contains the most dominant fur color, the big C, okay? So if you have the dark gray big C allele combined with another dark gray allele, with a chinchilla allele, with a light gray allele, or with an albino allele, it doesn't matter, you get a dark gray rabbit. Okay? The next most dominant is the chinchilla allele, and that gives you this nice soft gray color. But the only way you get a chinchilla rabbit is to have 2 copies of the chinchilla allele. Okay, so you have to have 2 copies of this sort of second dominant allele. Light gray occurs anytime you have one chinchilla allele and something more recessive than it, either the point restricted allele or the albino allele. Point restriction occurs anytime you have CH, a point restriction allele, and something the same or less dominant than it. We'll come back and look at point restriction in a little while. And then the least likely phenotype is the albino phenotype, where you have to have 2 of the most recessive alleles to be an albino. So depending on what combinations, if you breed a dark gray rabbit with a point restricted rabbit, right, you have all sorts of possible gamete combinations, and then that's going to give you what your potential offspring are. Okay. So most genes are more complicated than just 2 alleles. There are many alleles out there in the population, the global population of organisms, that can get passed around. So in this case, in these rabbits, we have 4 different alleles. How many alleles does each rabbit have? If there's 4 in the possible alleles in the global population, how many alleles does any given rabbit have for fur color gene? 2. It's not a trick question. Just 2, right? Because they have 2 genes that control fur color, 2 chromosomes with a gene for fur color. And they might have 2 identical alleles or 2 different alleles. But out of all the alleles in the population, any given individual can only have 2. Okay. So it doesn't matter if there's 2 total alleles in the world or 1,000 alleles in the world. Any given individual only has 2, whatever they inherited from their parents. Does that make sense? Pretend. Okay. The next thing that we have is codominance. So we have complete dominance, incomplete dominance, and then codominance. And what codominance means is that there are 2 alleles that are expressed equally when you have a heterozygote. The best example of this is in human blood type. Now, this is not the second part of blood type, the Rh factor, for which you're plus or minus. This is just A, AB, B, and O, the actual blood type. Okay, the plus or minus is separate. So those are the 4 possible phenotypes that you can have. You can be blood type A, blood type B, blood type AB, or blood type O, right? And which of those particular phenotypes you are is determined by a glycoprotein that you carry on the surface of your red blood cells. Okay. So if this is our red blood cell here, if you are type A, then you only have the A glycoprotein on the surface of your red blood cell. Okay, this is just a membrane protein. It's on the surface of the cell. If you're type B, you only have the B glycoprotein. Those individuals that are AB have both types glycoprotein. And those individuals that are O have no glycoproteins on the surface of their cells. Okay? It's the absence of that. Type O is the most recessive. A and B are codominant. Okay? So if your phenotype is to be type A, we always show blood type as an I, and I can't remember what I stands for. Immunoglobulin, I think. It's the factor that attacks the blood type. I, big A, means you have 2 copies of the dominant A allele, right? I, big A, little I, means that you have 1 copy of the codominant A allele and then 1 copy of the recessive O allele, the no glycoprotein. Does that make sense? Okay. Either one of these will give you a red blood cell with glycoprotein A on it. And excuse this diagram that says carbohydrate. It's more complicated than that. It's a glycoprotein. It's a sugar protein. Blood group B, in order to be a phenotype B, you either have 2 copies of the codominant B allele, or you have 1 copy of the dominant B allele and a recessive O allele. The only way you can be AB is if you have one A and one B, right? So you have to have one each of the two codominant alleles, and then you end up with both of those glycoproteins on the surface of your cell. The only way you can be blood type O is if you have two copies of the most recessive allele, the little I allele, which gives you no glycoproteins. Okay. Does that make sense? Because that has to do with Rh factor, which is a separate gene. You're not going to have to know all of the blood types, but well, you should probably know that A and B are codominant and that I is recessive. And like if I asked you to do a paternity test on your exam, you might want to be able to do that, right? Like mom has blood type AB, dad has blood type B, baby has blood type whatever. Is this possible? Yeah? All right. I was just making it up off the top of my head. It's weird. Okay. I always put this up here because people always ask. I'm not ever going to ask you to know this, but the reason that you can only have certain types of blood mixed together is because of antibodies produced in the blood. So individuals who are type A produce an antibody to type B. People who are type B produce an antibody to type A. AB doesn't produce any antibodies, and O produces both. So that means that AB is the universal recipient. They can receive any blood, type A, B, O, or AB. And O is the universal donor because it doesn't have a reaction with any of the antibodies produced by A, B, or AB. Okay, so this is just for fun because people always ask, which is universal donor, which is universal acceptor. I won't ask you that on an exam. Okay. The next thing that we have to mention is polygenic inheritance. And I kind of feel like that in all of the things that we think about in humans, like our physical features, if we're going to describe somebody, like what's on your driver's license, hair color, eye color, height, right? All of those things that we think about as human phenotypes are almost all polygenic inheritance, which just means that many genes affect one character. So your eye color is not the product of one gene, but it's the product of like a dozen genes, right? Your skin color is not the product of one gene. It is the product of over 378 genes, probably. At least 378 genes have been discovered that contribute to human skin color, right? So think about the continuum of skin color, right? It's a profound continuum from every possible combination. That's because so many different genes are impacting whether or not you produce melanin, how much of it you produce, how you deposit it, how fast you break it down. All of those things are contributing to your physical appearance, your phenotype, right? And so this is actually just assuming that skin color was a combination of 3 genes to look at a bigger Punnett square, right? And these are all the possible combinations of 3 genes. You multiply that out to 378 genes, we have a lot of possible combinations, right? Yeah. So many, many characters in humans and in all organisms are actually polygenic. They're a combination of multiple genes interacting together to give you your overall phenotype. That's only for 3 genes. Yeah, because 3 genes, you have 64 possible outcomes. If you have 378 genes, your total number of outcomes is much higher. Yeah, with most individuals in the middle. Yeah. I think eye color is around a dozen. I don't remember. I haven't looked it up recently.
Very basic. Blue eye color, brown eye color.
Yeah. Yeah. So there is one gene that is brown eye color and all other eye colors. So that is the one that is used most often. It's like brown and everything else. But that's not the only gene that contributes to eye color. That's just the brown versus other colors gene. I think it's around a dozen, but it's been a while since I looked it up. I hadn't looked up skin color in a while, and I was like, whoa, it's up to 378. Gotta update that because I think I had it like down in the dozens as opposed to hundreds. Because we're discovering more genes all the time and understanding what they do as we try to seek causes of disease and cures for disease and all those things. Yeah. Every time there's a mutation, you get a new allele, right? Technically. So all you need is one letter change and poof, new allele. Whether or not that allele actually does something different than the original allele. So like there's a functional difference versus actual difference, right? If it doesn't change the function, then we kind of don't count it as a change, or like in terms of the total number of alleles, because we don't have different possible outcomes. But yeah, anytime there's a change, you technically have the potential for a new allele. Riley?
So these are all laws that we're learning about?
Well, this isn't a law. This is just a type of inheritance.
Yeah, but
it's part of our laws of inheritance. Yeah.
So why are these laws of inheritance laws and evolution?
That's a good question. So, well, usually the difference between like the best definitional difference I found between theory and law is math. So a law is something that can be described mathematically, and a theory is something that has to be described qualitatively. And so if you can reduce it to a formula, then it can be a law. Like natural laws tend to be mathematically based. And so it may actually be the mathematical component behind genetics that causes it to be a law of inheritance versus a theory of inheritance. Okay, so the next type of inheritance is epistasis. And epistasis is something you've also already looked at. This was the extension in your lab handout, right? Basically, what happens in epistasis is that the phenotypic expression of one gene is influenced or controlled by another gene. So the purple seed color versus yellow seed color versus white seed color in corn was one example. Coat color in Labrador retrievers is another example. I'm a dog fan, so anytime I get to use dogs as an example, I'm going to use dogs. So the test cross example that I gave you had black labs versus chocolate labs. Black coat color in Labrador retrievers is dominant. Chocolate is recessive. Okay, so in order to be a black lab, you have to be big B, or big B, little b. In order to be a brown lab, you have to be little b for coat color, right? But in labs, coat color is controlled by another gene known as the expression gene. And so the expression gene controls whether or not the pigment that is produced is deposited. This is something else that contributes to human skin color, right? There's how much pigment you produce and then how effectively you deposit that pigment in your cells. So if you have a dominant allele for pigment deposition and you make black coat color, then that black color gets deposited in the hairs. If you have a dominant expression allele and you make brown coat color, that brown coat color gets deposited in the hairs. If you have 2 copies of the recessive allele for coat color or for expression, then it doesn't really matter whether you make brown pigment or black pigment. None of the pigment gets deposited in the hairs. And so a yellow lab is actually the absence of pigment deposition. The hair cells are empty of pigment, and so they appear yellow or white.
There is silver lab also.
Oh, I'm not familiar with silver labs. Okay. Like, that's too much. I'm sorry. I don't know about silver labs. I don't know about their genetics. Yeah. Yeah, I've never even heard of it before. I think we'd have to look it up. A dilution of.
It's a gene that affects like the degree to which like a color
An expression gene. Yeah. Okay. So it's probably somewhere in here, something polygenic about the expression gene. I'll have to look that one up and add it. I didn't even know there were silver labs. So this is what the F2 Punnett square looks like for a heterozygous black lab with coat color expression, heterozygous black lab with coat color expression, right? The majority of the individuals will have black pigment that is expressed, right? Some individuals will have black pigment that is unexpressed. That's these three here, right? Although they're producing black pigment, the two little e's mean they can't express it. Some individuals will have brown pigment that's expressed. And then 1 out of 16 will have brown pigment that's not expressed, right? So because gene E determines the expression of B, now our F2 phenotype ratio is no longer 9 to 3 to 3 to 1, but instead it is 9 to 3 to 3. Wait, no. 9 to 4 to 3. Right? Does that make sense? If you were struggling with your question on your lab, this Punnett square would help you with the white corn kernel gene. Huh?
I got that all wrong.
You still have until Wednesday to try again. I'm sorry about that.
Oh, sorry.
Moodle error. Yeah. Okay, so the next thing I want to talk about is outside of the genome, right? So we have all of these genes that can interact with each other, that can influence how things are expressed. We can have multiple genes contributing to one character, right? Beyond all of that, we also have the outside environment. So there was an age-old debate about nature versus nurture in psychology, right? Biologists will tell you it's not nature versus nurture, it's both, right? There is both the underlying genotype that you have, and then there is how your genes interact with the environment. And both of them are important in creating who you are, what your phenotype is, right? So it's important to remember that the environment is going to impact how genes are expressed. And when I say the environment, you can be talking about light, UV exposure, temperature, nutritional status, stress hormone circulating levels, right? And these things may be important in the adult organism. They can be really important in the developing organism, right? If you are an infant organism in utero and your mother is experiencing very high chronic stress, that may change your gene expression and likely impact you for the rest of your life, right? Some individuals in utero are impacted by temperature. Some species of reptiles become male or female depending on the temperature their parent is at, right? There are all sorts of things that happen to the genetic expression early on, especially. Okay, so we've referred to this as expressivity, the degree to which a particular genotype is expressed in an individual. So I have a couple of examples here. The first one is a Chaco tan. I'm pretty guilty of this in the summer. My skin color changes with exposure to UV light. My skin color darkens as I get exposure to UV light. It's because UV light is damaging to the cells, and the cells respond to that damage by upping their production of protective chemical, melanin. Right? So if you experience a tan when you're exposed to sun, that's because your body is trying to protect itself from that sun exposure. Right? Your hair lightens, it gets bleached. What causes bleaching? That's a good question. I don't know. Your hair, if it's just sun damage or if it's actually gene thing. I think it's actually just damage because only the hairs right at the very beginning are still alive, right? Everything else is dead. It's probably just damage. So sunburn is an extreme response, right? That's when you've really damaged a bunch of cells. And that will often cue your skin to up its expression of protective molecules, if it can, right? Some individuals can't produce any melanin. Some produce very little, And so your body will do what it can. If you evolve in a place where you're chronically exposed to a lot of sun, you will have naturally darker skin, right, to protect you from that sun. If you have very dark skin and you move to a place where there's very little sunlight, say northern latitudes, then you can't produce enough vitamin D. And so you have to have loss of some of that pigment. And so it's just adaptation to where you evolved, the amount of sun you're used to in your evolutionary history. These guys down here, the Siamese cat and this funny looking rabbit, sorry, I think it's funny with its big eyes, are both exhibiting what we call point restricted color. And so point restriction was one of the color types when we were looking at rabbit fur. It just means having dark extremities and a light body. So this cat has a light body, but it has dark ear tips, dark nose, dark tail, and mostly dark paws. The rabbit is very clearly dark nose, dark ears, dark paws, dark tail, and the rest is white. It turns out that the pigment that is produced in this rabbit's body, the dark pigment, is produced by an enzyme that denatures at high body temperatures. So the rabbit is producing dark color in all of its body, but its core body temperature is high enough to denature that pigment, so it's not expressed in the core. It's only expressed in the extremities. And this example goes so far to be so cool. If you shave the back of this rabbit right here, and then apply an ice pack, like one of those cold circulating ice packs, if any of you have ever had orthopedic surgery, strap a little perpetual ice pack onto them. While the hair is growing back, the hair grows back black. It's totally temperature dependent. And so that color is just dependent on the temperature of the body. Because the core is warmer than the extremities, the core ends up being a different color than the extremities. You totally could make a checkerboard rabbit. Associated with expressivity is the idea of penetrance. And penetrance is the proportion of individuals with a genotype that actually express the phenotype. Okay? So expressivity is the degree to which you express your genotype, and penetrance is the proportion of individuals that show the typical phenotype. Okay? And I have Woody Guthrie up here because Woody Guthrie developed Huntington's disease. Huntington's disease is an autosomal dominant disease, which means you only need one copy of it to have it. It was much more prevalent before we understood how it was inherited, because most individuals don't actually develop the disease until they're well into their 30s or even their 40s. So historically, that was well past childbearing years. And so individuals would have already passed the gene on to their offspring before they even knew that they had it. With awareness, many families that had Huntington's in their family actually chose not to reproduce because it's a pretty traumatic way for an individual to decline. There's really no treatment for it. It causes atrophy of the brain. And so this is down here, normal brain. And this is a brain on Huntington's disease. Huge quantities of tissue just lost. Right? So you lose your cognitive capacity, you lose motor function, you end up spending any number of years in a pretty incapacitated state before it ultimately results in death. What we found out as people who had a history of Huntington's disease in their family wanted to know, do I have the Huntington's gene or not? Should I have kids or not? Is that 5% of people who have the allele never get the disease. So Huntington's disease is 95% penetrant. 5% of the people might have the allele and never show any sign of disease. So it makes it even more complicated to make those kinds of decisions in a genetic counseling kind of situation. But it's just like, we didn't know that was possible until we started having the tools available to do so much genetic testing and discover that people who have this allele, we can see it in their genome, aren't expressing it, right? What else is going on? Maybe there's some other gene that's turning off the Huntington's gene, right? And I'm sure that that's an active area of study. The last time I checked, we had not figured that out at all. I checked about 6 months ago. So. Woody Guthrie's situation is really, really funny. His children from his first marriage had it, and his children from his second marriage did not, which doesn't make any sense. It's got to be totally random chance, but that's how it worked out. So his older children were affected. His younger children were not. Whoops, wrong way. There's lots of things that destroy brain tissue. Alzheimer's actually produces a plaque protein that builds up and cuts off, I think it cuts off the circulation to the tissue. So ultimately the tissue dies, but it actually comes first from these proteins, I think. Proteins called plaques. Non-Alzheimer's expert, you should check it out. I'm going to stop here because the next slide starts a section that's more than a minute long. So we will finish just the last little bit of chapter 10 and start chapter 11. I'll get chapter 11 up on Moodle. Lab 11 for Wednesday is up, and there is a small PowerPoint that we will go through in lab that is a supplement to the lab. We're going to do everything that we do about DNA biotechnology in lab on Wednesday, and that'll be it. But the material from that PowerPoint will be part of our last exam. That's a good question.
Like